Good morning, students. Myself, Mr. Sriharsha Vandavasi from Assistant Professor, Indian University, Petroleum Department. So today we are going to discuss about health, safety, and environment. So today's our topic is impact of drilling and production operations on environment. So how the drilling and production operations are impact on the environment. So this is our topic name. So we will go brief into the topic. So subtopic of the today's So the subtopic of the today's session is measuring toxicity. So what are the contents today I will going to discuss. So today content is how toxicity, what is toxicity, and measuring of toxicity. Next, bioassay, how we will measure the toxicity by using some methods like bioassay and limitations. And next after that we will go with the microtox method, which is used for the toxicity measurement. So these are my contents. So by end of this lecture, you are going to understand these four topics. First one is toxicity, second one is toxicity measurement, third one is bioassay and limitations, and the final one is microtox method. So these are four topics we are going to discuss today. So toxicity. So first of all, when it comes to the toxicity, so all of you feel toxic means it is a harmful material. So by the name itself, you can say toxicity is the substance which is going to harm an animal or the organism. So this one is called as a toxicity. Any substance which causing a harmful effects on a living organism we call it as a toxic material. So how much the substance the substance at which grade it is going to disturb the organism, and how much so in this, so the degree at which the microorganism is affected by the toxic material or the substance we call it as a toxicity. So we will start with the introduction of toxicity. So I already told you, so the degree of damage that is caused by a material or a substance to a living organism, we call it as a toxicity. So next, so how can we measure the toxicity? So we will measure the toxicity by using laboratory methods. So what we will do, we will take some organism or laboratory organism, and we will test the organism by using the substance. So according to the substance, how much the organism is affected, that we calculate and that we analyze. So according to that, we will measure how much effect is imposed on the organism due to that certain substance. So this is called measuring of toxicity. So we will do the lab test by using the same substance on different organisms. So generally what in petroleum industry waste, so in petroleum industry we are using a different toxic material. So in generally what we will do, so the common animals or organisms which are affecting there, we will test it in the laboratory so that we can tell how much effect is given by the toxic material. So here in petroleum industry waste, we use mainly two types of things. One is the marine organisms, another one is the freshwater organisms. So what the our operations are everyone going on, drilling and production, everything. These wastes are we are going to dump in the freshwater, or otherwise in the marine water. So suppose if you are dumping the fresh water, so how much it should be the toxic levels? And suppose you are sending in the marine water, then how much it is the toxic level? So according to that, by using the different samples, then we will calculate it. So here I mentioned you the petroleum industries. So in marine water we are using two things. One is mason trim and fathead minion. So next one is in freshwater we will use damp-head shrimp and fathead minion. So these are the two things which we are using in the petroleum industry based calculation of the toxicity. So there are two types. One is marine water, another one is fresh water. In marine water we are using damp-head shrimp and fathead minion. And at the same time in the fresh water we will use marine mashed shrimp and shrimp-head melon. So these are the two things. These are two samples species we are going to test during testing of the toxicity. So generally, what is the symbol of the toxicity is, you can see that the toxicity symbol like bone with the cross bones with the skull. So here I will show you the diagrams of this is the dam head ship. This one we are using, and this is the fat head minnow. So these are the things which we are using there. So these are the two samples which we are using for the testing purpose. So two species. So the freshwater we are using dam head shrimp and fat head minnow, and marine water we are using mice head shrimp and sheep head minnow. So next toxicity measurement. So how we will measure the toxic? So we know that what are the toxic, what are the species which are using for the laboratory purpose for measuring toxicity. So what are the methods of measuring the toxicity? So here we have the toxicity measurement techniques. That means how much toxicity is going to affect on organism. So here I mentioned the toxicity. So toxicity again it is divided into two types. One is the dose, and one is the concentration. So in the dose also we have two subparts like LD50 and LDLO, and the concentration you have LC50 and LDLO. So these are the criteria. So we will further go into the topic and we will discuss what is that. So what dose? The dose means the mass fraction of the substance which is present in the living organism. That is the toxic material which is already present in the organism because of inhalation or digestion or something like injection, something like that. Any different process, the organism is affected with the material. The material is already in its body. So this is we call as a dose. This we will calculate according to the mg per gram. So how many milligrams of the substance is present in the gram body of the animal. This is called the dose. Here you can see. Next one is the concentration. Concentration means concentration means it is a large quantities, right? So in the concentration, what we will do, the amount of the substance which is present in that environment. That means the substance already is in the environment. In that environment, if you are sending any animal, then how much effect it is going to be imposed on the animal. So this is called the concentration. So concentration generally speak in ppm, parts per million. You can take it as a mg per liter also. So milligrams of substance which is present in the liter of water or liter of that solvent, something like that. So how much concentration is present in that particular environment, we call as is called as a concentration. So we're coming to the dose. So we already discussed before, we discussed about the dose LD50 and LCLO. So what are the LD50 and LCLO? I will discuss right now. So LD50 means the lethal effect which is going to die 50% of the animals. So 50% of the animals we are going to die during this dosage that we called as a LD50. Because of this dosage, the 50% of the animals have the lethal effects, that means harmful effects, that we called as a LD50. Then what about the LDLO? The lowest amount at which the first depth of the animal will be, is going to be faced. That means LDLO. So next one, the concentration. So the organisms which are in that concentration, which we have in 50% of the lethal concentration, the lethal concentration which is going to kill 50% of the animals in that environment, we call it as an LC50. And what about in the same concentration which is present in the organism for a particular given time, so that the least lethal effects, that means starting, the lowest lethal effects will be going to be formed. That is called LDL, that is called LCL. So these are the two things. One is the dose, another is the concentration. So first one is 50% of the animals which are going to be affected, that is LD50, and the first animal going to die, that is LDL. And if you come to the concentration, the first 50% of the animals which are there, which are affected by that concentration. So that is called LC50. And the same particular time period, if you done the same test, but the lowest lethal effects, that means just the lowest lethal effects which is formed on, which is going to affect the organism, called LCLO. So these are the LC50 and LCLD, those are concentration. Then we go further into inside. So next one is length of exposure of substance in a descriptive ways. So suppose we have a substance. So how much time it is the exposure to that particular time? Suppose we having the different toxic materials. So you can ask me, sir, which type will be gives which time of effect. So suppose you are in an acidic zone. Okay, then automatically suppose it fell on your hand. within 24 hours. That means within the seconds only it will react on your body. So that is called acute effect. That means according to the time duration, the substance effect on the human being, that we call it as a that we call it as a acute. That means that we call it as a different length of exposure time of exposure. So suppose it is happened before them. So first one is acute. So different ways means, so acute way. So in acute way, what will happens? In acute ways, suppose anything, the substance fell on your body, which will give impact within the 24 hours. That means it will give the results in the 24 hours. Suppose acid is fell on your body, automatically it's totally everything burns. So it is less than 24 hours. That means less than 5 minutes or like that. So this is called, comes under acute length. Next one is when you go for the subacute. That means it takes some time more than the acute. So what happens here is it takes some time for the giving the reaction. Suppose you see if you use some chemicals. Okay, suppose you are using the same chemical for the more than 1 month or like that. So first 1 month it is there is no that much difference. But after some time, what happens slowly, the itching or irritation will comes on the body. So that is called subacute. That means within the up to less than up to 3 months or something like up to 1 month, up to less than 1 month, it is going to affect on your body. That is called subacute. So that the exposure time is less than 30 days. So next one is called subchronic. Subchronic means you are in a zone, like you can say the exposure time will be like it will be exposure time up to 1 month to up to 3 months. So that is like up to 90 days period of time. So that one we call it as a subchronic. So subchronic are like you are in a air pollution or you are in a sound pollution like that. So it will not affect you in a short period of time. It will be actually shown after later some time. So this one we call as subchronic. The final one is chronic. So it will be like exposure is more than a year, more than 3 months. So chronic means the exposure. Chronic means the exposure is more than 3 months. So in these 3 months, more than 3 months means you can say these are like your chemical carbon elements, carbon dioxide, carbon monoxide. So these type of elements you are exposing for more than 3 months. So automatically it will lead to the cancer. So cancer type of cancer type of disease which are formed because of exposure more than the 3 months, then we call it as it's a chronic disease or chronic substance. So next we will start with the bioassay and limitations. So what is bioassay? So bioassay means it is a laboratory method. That means laboratory analysis. Actually, bioassay means inside phone you can say biological analysis. So it is the biological analysis which we are going to do in the process. So during bioassay, what will happen? In bioassay, we will take sample and we will keep it in the laboratory with the substance. So during the laboratory issues, what happens? The impact which is going to be give a substance on the organism or a microorganism. It takes some time, and after some time, it will give the result. So in bioassay, what we will do? We will use some laboratory equipment and the laboratory substances. So in that, what we will keep is we will keep them with the microorganisms in that the same laboratory in the same equipment, and we test it. By testing it, what we will know, we will know that how much effect it is going to give it on the microorganism. So according to these effects which are formed on the microorganism, we will prepare a drug. So that is called as a bioassay. So bioassay has some limitations. The limitations also we will discuss right now. So coming to bioassay, it's a biological assay. Means so it is conducted on the measures of it is conducted on an organism to measure the effects which are given by the organism because to the substance which we are using there. So limitations, coming to the limitations, the test yield is going to give the acute. So acute lethal concentration. That means there takes acute period of time. That means less than 24 hours only the results will be given. So that also they will conduct this test for the lethal concentrations. That means very harmful concentrations only they will conduct. Next one, they are conducted in the laboratory but so the same thing, it will not be applicable to the field search. So suppose you are using in a field study, like you are in a working zone, then you can't apply these things there. So there you can't apply. So this is only limited to the laboratory purpose. The field work or the field results may be vary from each other. So next one is, so during this bioassay, so it is conducted in a short period of time. So they don't give the adequate information about the carcinogenic or mutant effects. Suppose some carcinogenic effect is there, or mutation changes, mutant alteration is there, that effects will not be observed during the this bioassay method. So why? Because of this is lethal and it is happened in acute method. So the time period is very less, so we don't get adequate information about the bioassay. So one more limitation of the bioassay is the time period. So within 2 to 3 weeks, you will get the results of the bioassay. So next one is the microtox method. This is also one type of method which is useful for the checking the harmfulness of the substance on the organism. So in microtox method, what we will do, we will use the microtox method. We will use the luminescent bacterium. So this bacterium will emit a light. So while we are keeping this bacterium with the substance in a confined space. So what happens? The substance will affect on the toxic material will affect on the substance, will affect on the microorganism. So slowly what happens? This is a luminescent bacterium is going to emit out the light according to the material which is affected. So suppose effect is more on the bacteria, automatically the light emission from the bacteria will become less. So according to that, how much impact is there? According to that, we will decide the toxic levels in the toxic levels of the material. So during the process, what happens? The metabolic process of the organism will disturb. So according to that substance, and it will not perform its own activities properly. That means like it's breathing, it may have a breathing problem, like inhalation problem, a digestion problem, or the injection problem. So these problems may occur during this process. So during this process, the substance will impact on the metabolic process of the organism. So finally, the reduction in light gives you the how much effect it is giving on the organism, and that we will consider the toxic material which is present in the substance. So what are the main advantages this test is? The results will be conducted will be the results will be declared within the 15 minutes. Thank you.